Rajan sir.
Shall we? ಶುರು ಮಾಡೋಣ.
Rajan sir.
ಚಂದನ್.
Yes, join ಆಗಿದ್ದಾರೆ already.
Oh, all right. Fine. Sir, shall we, sir?
Yeah, the resource person is ready, yeah?
Yes, sir. He has already joined, sir.
Yes, I'm here.
Okay.
He's already joined, sir.
Good evening, Dr. Deepak.
Hi, good morning. I'm sorry.
Good morning. Good morning, Dr. Deepak.
Yeah. Good evening.
Yeah, good morning, Dr. Deepak. Welcome, welcome.
Oh, thank you. Thank you. Nice being here.
A very good evening to all the participants. I am Professor Rajarajan, Head of the Department of Microbiology, Indian Academy Degree College, Autonomous, Bangalore, India. At the outset, I would like to invite all the participants who have shown really acute interest in registering in the seminar on bacterial virulence, perceptive and pragmatic approach. I feel it is up to the fate in the resource person and the institution that we have received a tremendous response in the form of 3,000 plus registrations, which we have received from more than 15 states of India and 8 nationalities. We are really proud of it. At the same time, Dr. Deepak Balasubramaniam, the resource person of the day, was very, very kind enough to share his time and expertise to the entire microbiology community. And in the advent of the time when we are really experiencing a situation where and when microbiology is really going to play a pivotal role in the day-to-day life of people, and the whole world has been going through a tough and hard time. It is really understandable that people should realize why microbiology is a very, very important part of our life. With that, I would like to invite our beloved principal, Dr. E. Jerome Xavier, to address the participants and welcome them.
Good evening, dear academicians, scientists, research scholars, and you, Dr. Deepak, are resource persons for this evening. For an intellectual and inquisitive mind, space, time, and situation has no restriction, for the process of learning cannot be stopped to such minds. The pandemic situation that we are forced to face with has created a beautiful and fantastic opportunity for
Yes, sir. Yes, sir. We do, sir.
Created a fantastic opportunity for the community of academicians, research scholars, and scientists all over the world. I am extremely delighted to have over 3,000 audience. Normally I call them fraternity of intellectuals who are ready to learn and leap forward in any given situations. A very warm welcome to each one of you who have joined us this evening. I do appreciate the initiative and the trouble taken by the Department of Microbiology under the headship of Professor Rajarajan. Indian Academic Degree College is an edifice of learning which is built on a spectacle of fine blend of tradition and the ever-changing modernity. The hallmark of every eminent higher educational institution is quality and excellence it provides in nurturing the young minds and influencing the molding of character. The systematic process of education, set in an ambience of learning, stimulates our students to acquire academic excellence along with co-curricular and extracurricular activities to have a holistic development. Its modernity is reflected through the introduction of career-oriented programs in physical and life sciences, commerce, management, and humanities. The integration of academic excellence, developing skills and competencies, and professional learning are the unique strengths of our institution. It is the collective efforts of management, staff, and students that make this college such a vibrant place to learn, to work, and to explore. The reputation of our faculty, the quality of our academic programs, the personalized attention and plethora of co-curricular and extracurricular activities draw students from 25 states of India and 17 countries. This speaks of the rich student diversity at Indian Academy. Challenged by outstanding faculty and an outcome-based curriculum, students are well prepared to contribute to society and to be effective leaders of social change based on the ethical values and principles the student acquire from the college. The college has scaled new heights of glory coupled with widespread achievements in the spheres of cultural and curricular activities. On behalf of the management and the faculties of microbiology and the rest of the staff members of Indian Academy Degree College Autonomous Bangalore, I extend a most hearty welcome to every participant to this webinar on Bacterial Virulence, Perceptive and Pragmatic Approach, which will be delivered by Dr. Deepak Balasubramanian. Thank you. Once again, I extend a very warm welcome to you all.
Thank you, sir, for those kind words. I would like to now I would like to introduce the resource person of the day, Dr. Deepak Balasubramanian. Dr. Deepak had his Bachelor of Science in Microbiology from the University of Madras. And in fact, of course, me and Dr. Deepak both are alumnus from the same college.
That's true.
Yeah, thank you. And he went on to pursue his master's from Madurai Kamaraj University. And further ahead, he went to Florida International University to obtain his PhD in molecular microbiology. Later, coming back down to India, like he started his career as a lecturer at his alma mater, that is MGR College, Hazore. Further, he went on to join as a senior research associate at the Bangalore Genie Private Limited, now being called Bangalore Genie. He has served there for 3 years, and he was instrumental in developing a lot of novel techniques, novel therapeutic techniques there. Further ahead, he joined Gangajan Biotech, Bangalore, India, where he was further again instrumental in developing bacteriophage-based products for human therapeutic applications to deal with multi-drug resistant bacteria, particularly MRSA and MDR Pseudomonas aeruginosa. From there, he went on to join Florida International University to pursue his PhD. Back to India in 2013, he was he was associated with the Florida International University itself for another 4 months in developing in silico analysis techniques to identify non-coding RNA targets in bacterial systems. Dr. Deepak had a long and he has a very, very long and enduring research career. And the hallmark, the highlight of his research career came further in 2013 when he joined Harvard Medical School in Greater Boston, where he was instrumental in developing in vivo assay system for performing structural function analysis of interactions between non-coding RNA and their mRNA targets. Now, followed by November 2015, he came back to India in February 2016 and joined as a principal scientist in his uh, company again, where he was serving earlier, that is at Gangajan, Bangalore, Gangajan Biotech Inc., Bangalore, India. And that was a very fruitful time for Dr. Deepak, wherein he developed a lead molecule targeting Pseudomonas aeruginosa and related bacteria, resulting in a US patent, omics to approach identified target bacteria for environmental phage application studies. And he has also developed a strategy, and he designed as well by validating it with for chimeric proteins based animal bacterial agents for escape pathogens, that is Enterobacter, Staphylococcus aureus, Klebsiella pneumoniae, Acinetobacter, Pseudomonas, and other Enterobacter pathogens. And that's how he has a very long history of research career. From there, he went back to US to join Vanderbilt University, one of the prestigious universities in US, as a research fellow. From there, he came back to India again in September 18 and joined Public Health Research Institute of India in Mysore. And from there, he went back to US again, to University of Central Florida, Orlando, where he's pursuing his research since July 2019 onwards. Dr. Deepak is decorated with 14 spectacular publications, journals in a highly reputed, highly reputed journals, and he is also a life member of American Society for Microbiology, shortly known as ASM. Dr. Deepak also has a patent to him, which is something towards the invention which provides methods and compositions to reduce growth of microbial colonies, including infections, and includes therapeutic combinations, methods for treatment of infections, and methods for identifying additional such composition. So we are really, really happy and overwhelmed by your consent, Dr. Deepak, and we are really happy to have you with us this evening. It is over. It is over to Dr. Deepak to start your presentation.
Sure. Thank you, Dr. Rajarajan, for those kind words. We go a long way back. I haven't seen you in so many years now that I realized suddenly that you are at Bangalore and here. But anyway, thank you all for inviting me. It's been a really a pleasure. And Malay and me, Dr. Malay Sapandi and me too, we go a long way back too, because we did our masters together. What seems like ages ago. So today I'm going to be talking about. Let me start this screen share.
So just 1 minute, sir. We are just having some internet issue. We'll start in another 1 minute.
Sure.
Yes, sir. Just for 1 minute.
Okay.
Karan sir, just receive my call.
Yeah. Yeah. Yes, Krishna? Hello.
Rajan sir,
Yes, sir.
they are reconnecting to the other one. 1 minute they require because the
Yes, sir. Yes,
Facebook
sir.
uh,
Yes, sir.
Facebook audio is not quite okay. Mr. Chandan is doing it, so 1 minute they require. Just they'll do it.
Yes, Krishna. Yes, yes, yes. Right. Just a couple of moments, dear participants. We are just connecting you online live. So there is always a gap between Facebook streaming and hardly few seconds between the real streaming and the online streaming towards the Facebook. So kindly bear with us.
So you could connect, sir. I couldn't contact them because all phones are engaged. Again, you just what I did. No, I joined the link. Then Cisco meetings. It is opening, showing us Cisco meetings. Webex. That in the mail join the link. Alarasa Pandin sir has forwarded. So in that, when I clicked that join the link, then okay. Lata ma'am.
I don't know why. No, why most of the technology videos and all those pathogens?
Chandan, ಆಗಿದೆಯಾ?
Sir, we are ready, sir.
Yeah, over to you, Dr. Deepak.
Sure. So, again, thank you all for coming, and thanks for the invite. Today we're going to talk about bacterial virulence mechanisms. But given the vastness of the topic and given the obvious time limitation, I'm just going to give you like an overview of what bacterial virulence means, and then we'll get into a little bit of practical approaches in trying to study bacterial virulence. Now, the WHO announced in 2017 that about 40% of the deaths worldwide are contributed by infectious diseases, which is a very serious issue. And that, again, is going to get even worse because we'll have, by 2050, over 10 million people that are going to be killed by antimicrobial drug-resistant infectious diseases, which, alarmingly, is going to be much higher than the next two major killers projected, cancer and diabetes put together. So it's imperative that we address this issue now, or at least start addressing this issue now, in order to, to try and lower this number as much as we can. Now, this is in spite of the fact that of the 10 to 100 million bacteria that are known to exist, not just. We have only about a few hundred of them, which is way less than 5% of the bacteria that are human pathogens, which begs to emphasize that virulence is a rare exception, but it's not the rule. Bacteria are not out there to kill you. They are just doing their own thing, and we just happen to be hosts, and we'll talk a little bit about that later on. But in spite of all these statistics, these are just numbers. The bottom line is that unless we study bacterial virulence, we will not be able to address this very serious issue of antimicrobial drug resistance or saving human lives. And studying this phenomena of bacterial virulence will have design intervention strategies. Now, the concept of virulence, I'm trying to get rid of this thing. Anyway, the concept of virulence has been defined as the relative capacity of the microbe to cause the disease traditionally. But this is, I mean, people are now realizing that this is a very host-centered approach, so which means that if the bacteria has virulence factors or not, and then it's either classified as a pathogen or not. But that again, like I said, is a very host-centered measure in the sense that there are exceptions to this rule. For example, an organism which is classified as a pathogen need not always behave as a pathogen, and there is a very big host component to this process. I'm sure you've all heard of Typhoid Mary, who, in spite of being a carrier of a pathogen, she was just that, a carrier. She never displayed any symptoms, but she was able to spread the disease to numerous, hundreds of thousands of people, resulting in their deaths. So what that means is that in spite of being a pathogen, that particular strain of bacteria was not able to cause disease in that particular person. And that's just an example. There are numerous examples all over, including in COVID-19, where you have asymptomatic carriers. On the flip side, you have opportunistic microbes or commensal microbes, both opportunistic microbes and commensals, which in a healthy host are not able to cause the disease, but in an immunocompromised host, they can cause the disease. So all this goes to show that the traditional definition of virulence, which is the relative capacity of the microbe to cause the disease, which is a concept that is limited to just the microbe, and it doesn't take the host component into picture. But by and large, we are now realizing that the concept of virulence is a dynamic and complex phenomena which encompasses both the host and the microbial factors. So irrespective of these, we can talk about the definition of virulence all we want, but the bottom line is we are still being, the pathogens are still able to cause disease. And that is primarily mediated by virulence determinants. Now, these are pathogen-encoded mediators of infection, and there are two major types, depending on what the pathogen is. They can either be requisite virulence determinants, where they have a single primary virulence factor. Examples include capsules of Streptococcus pneumoniae or the Corynebacterium diphtheria toxin, where the presence of this factor alone is a determining factor whether the bacteria is able to cause disease or not. Right? So it's one factor, one virulence disease, one disease phenomena. But by and large, most of the bacteria do not belong to this category. Sure, there are a handful of them, but then most of the bacteria have a multifactorial effect. What I mean by that is we don't have a single virulence factor that you can attribute to that particular bacterium and say that this is the reason why this bacteria is causing disease. It's all contributive effect. You have proteases and phospholipases of Pseudomonas aeruginosa, which are required at specific stages of infection, but they are not always expressed, and they help in the infection process, but they are not the sole contributors of virulence. Similarly, toxins and adhesins of Staph aureus, they all help in the infection process, but they are not the only virulence factor that the bacteria possesses. But based on a timed expression in a method by which bacteria senses the environment, a plethora of virulence factors can together contribute to the pathogenesis of the bacteria. And that's what multifactorial effects are. Again, the caveat here is that in case of hosts with immune defects, like in immunocompromised patients, cancer, AIDS patients, or with underlying conditions such as cystic fibrosis, even an opportunistic pathogen, which is multifactorial, where individual virulence factors cannot cause the disease, can still cause the disease. That again goes back to what I was talking about earlier. Now, the traditional bacterial virulence determinants can be either cell-associated, such as the flagellum or the pili, or even LPS, lipopolysaccharides. They can be inherent properties of the bacterium, inherent or acquired properties of the bacterium, such as antibiotic resistance or quorum sensing, or they can be extracellular, such as outer membrane vesicles, proteases, hemolysins, toxins, and various things that are secreted by the various secretory machineries in the bacteria. Now, we don't have time to go into all of these virulence determinants, so I'll just touch upon a few of these major important ones. But if you have time, I'd strongly encourage you to go read about outer membrane vesicles, for example, which is a fascinating story of how these bacteria are able to communicate with each other, exchange genetic information, proteins, virulence factors, and this is a major contributing factor in cell-to-cell communication and in pathogenic determinants in communities such as in biofilms. But for today's talk, we'll focus on a few others, such as, and we'll start off with flagellum and pili, which are bacterial appendages. Both of these, again, contribute to the virulence factors. They are not the sole determining factors, but they both contribute to locomotion, colonization, and other virulence mechanisms. For example, the flagella is required for motility in blood, biofilms, or mucus. So they basically help the bacteria reach the site of infection. But once they reach the site of infection, their expression is typically shut off, and other virulence factors take over. So this sort of spatial temporal expression of virulence factors is very common, and it's a common theme among bacteria that have a multifactorial virulence mechanisms. Now, once they reach a solid surface, like in tissues, bacteria are still able to move around, but then they don't typically use flagella-mediated motility, but they switch over to a type 4 pili-mediated motility, which is like a grappling hood mechanism. Now, again, this, like I mentioned, depends on the stage of infection. What I mean by that is, for example, this is the case of Vibrio cholerae, which uses swimming motility to, so once it enters the intestine, this, what you see here is the intestinal epithelium with the microvilli shown here in blue. But then the bacterium, once it reaches there, in response to external cues, in this case, bile and bicarbonate, there are sensing mechanisms in the bacteria. It allows the bacteria to use a flagella-mediated motility to swim through the mucus layer and attach using non-specific adhesins to the cell surface. And once that happens, then this is the spatiotemporal expression I was talking about. The bacteria switches off flagella biosynthesis, well, actually in the next stage, but it starts producing site-specific adhesins, which are shown here in red. And once that happens and the bacteria start to colonize better, they turn off flagella biosynthesis, start producing type 4 pili. So this is the initial colonization process that the bacteria and Vibrio cholerae uses before it produces toxins, the cholera toxin, and causes a disease. So again, this is the initial colonization process in which flagella-mediated swimming motility is critical. And people have demonstrated that flagellar mutants are not able to initiate infection in Vibrio and in other bacteria. Now, once they reach the surface, type 4 pili and the mediated twitching motility becomes very important. What you see, and these are very important, especially in microcolony formation, which is part of the biofilm process. Now, what you see here are twitching plates where, based on from the initial site of inoculum, you can see that the wild type strain is able to twitch to a much greater extent compared to the pili mutant, which is not able to twitch at all. Now, the reason I'm showing these plates specifically is to show that some of these assays for virulence factors are very easy to perform with very minimal requirements in the lab, but they give you very meaningful data on the pathogenesis and in the virulence mechanisms. And this is especially useful for for master students who have their projects going on. If you want to analyze your isolates, these are some of the simple assays that you can do and get very meaningful information out of it. And I have a few more coming up, and we'll talk about that when we talk about the particular phenotype. But getting back to this, swimming and increasing motility are involved in the initial colonization process, whether it be in the infection or in microcolony formation, for example, in biofilms, which is another major virulence determinant. Now, in case you're not aware, biofilms are surface-associated microbial communities. You can think of them as microbial cities. So bacteria don't like living alone. They also are social beings in that sense. So what happens is that whether in the environment or in the process of infection, most bacteria form microbial communities, which are surface associated. They like to stay put and cause a disease. Now, this is seen in both acute and chronic infections, more so in chronic infections, but it's also being seen in acute infections depending on the bacteria. And like you see in a nice cosmopolitan city, like in Bangalore, for example, you have people of many different ethnicities living together. Similarly, microbial biofilms are also polymicrobial. You can, any situation you take, you will not, at least we don't know yet. We don't have any examples yet of a single a monomicrobial biofilm. It's always been polymicrobial. So the process begins with a flagellar-mediated motility attachment to either biotic or abiotic surfaces by planktonic cells. And once that happens, flagellar biosynthesis is shut off, and then they start dividing locally and producing this extracellular polysaccharide shown here in yellow. And this stage is called the microcolony formation, and this is the initial process of biofilm formation. Once that happens, the biofilm starts developing. There's layering, there's gradation. People have showed in elegant experiments that there is nutrient and oxygen gradients in the biofilm, and that is a major driving force in determining the physiology of the bacterial cells. And then that again develops into mature biofilms, where you see these huge microbial communities surrounded by extracellular polysaccharides. And there are lots of changes that happen to the cell when it decides to grow as a biofilm, because it's a major lifestyle change for the bacteria to go from being single cell planktonic to being live in a community like this, crowded. It's like being in a rural area versus being in a major cosmopolitan city. You adapt and change. Similarly, bacteria do the same thing. And under certain conditions and in response to certain cues, bacteria from biofilms can start expressing flagella again. They are dispersed out, and they can go reinitiate the process somewhere else. Now, cells grown in a biofilm undergo extensive changes in transcription and physiology. Like I said, it's a major change for the bacteria. So the gene expression completely changes. The physiology of the bacterial cells change. They start producing a very energy expensive extracellular polysaccharide. Alginate in Pseudomonas aeruginosa, you have various different kinds of polymers which are produced in other bacteria. Their metabolism goes down, and that is because not only because of lower nutrient availability, but then again, like I said, because of changes in transcription and the reduced metabolism of the cell, they become more refractory to antibiotics, because as you all know, antibiotics work only on actively dividing cells. The same thing with other chemical agents. Cells grown in biofilms are more chlorine resistant, and to other antimicrobial agents. There is also an enhanced stress response. There is significant upregulation of RPOS and other stationary phase stress response signal factors. And like I said, it's also found in both acute and chronic infections. And this is a recent paper that came out, I think, last week, which I found very interesting, which very clearly demonstrates that compared to planktonic cells, biofilm-grown cells are much more competitive in colonizing the intestinal epithelia. Whether you grow it on various surfaces, it doesn't matter whether you grow it on biotic or abiotic surfaces. Overall, biofilm-grown cells have a higher competitive index in the sense that they're able to outcompete planktonic-grown cells and able to colonize the intestine. So this clearly, I mean, they go on to do much more elegant studies in this paper. But anyway, but what this means is that biofilm cells are bad news for the patient, and biofilm is a very critical phenotype for the bacteria to be able to cause an infection. Now, going back to the assays, which students can do very quickly in the lab, a biofilm assay is a simple assay that can be done with very minimal equipment. Of course, there are other advanced ways of doing this, but you could basically just simply dilute an overnight broth in 96 fuel plates or tubes, incubate for varying time periods, and then quantify with crystal violet. And you will see that. So if you do an assay like this and you see a result like that and you don't know what the genotypes of the strains are, this is the wild type strain, for example, which forms some biofilm. This is all in Pseudomonas aeruginosa. You'll see that there are some biofilm which is formed, but then a negative control, which is a triple deletion mutant, is not able to form a biofilm, whereas 2 other mutants are able to form biofilms. So what this means is that if you don't have information on the strains, and you look at just this data, you will be able to say that, you know, there is something going on with this strain which does not allow it to form a biofilm versus this strain or these strains which can form a biofilm. So even in the absence of any genetic information on these strains, by the way, these are the negative and the positive controls, and these are the 2 test strains. But you will be able to identify a lot of information on the strains in terms of its virulence factor production by doing very simple assays. But a lot of these virulence factors are all co-regulated by a process called quorum sensing. Now, this is the way by which cell-to-cell communication happens in the bacteria. I like to sometimes tell my students that, you know, quorum sensing is like Twitter for the bacteria. If you alone go shout out in the street about some cause that you're concerned about, nobody's going to give a care about it. They're going to call you crazy. But then if you send out tweets, get a bunch of your friends, you all go together and protest in the streets, then you're going to have an impact. Similarly with quorum sensing, a single cell expressing a virulence factor in the host is going to be wiped out by the immune system. But then if you lay quiet, communicate with your neighbors, and then coordinate your gene expression together, then you have a much better chance of success. So that's what the bacteria do. Quorum sensing is a way bacteria talk to each other and coordinate their gene expression. And we know that there are multiple virulence factors, including biofilm formation, which are controlled by quorum sensing. And quorum sensing dictates community behavior. And the way bacteria talk to each other is by using chemical messengers called quorumones. The kind of quorumones that the bacteria use differ depending on the type of the bacteria. Gram-positives have peptide-based quorumone sensing, whereas gram-negatives are homoserine lactone-based, or in this case of Pseudomonas, there's an additional quinolone-based quorum sensing system. And the way quorum sensing system works is that in when there's a low population density, quorumones are they're constitutively expressed by these genes called synthetases. And the name of the synthetase depends on the kind of quorum sensing you have. They produce these chemical signals called synthetases, which are secreted out into the environment of the bacteria, and nothing happens. So at low population densities, they're just secreted out. But then as the population density increases, the bacterial chatter also continues to increase. And then these quorumone molecules diffuse back into the cell. In some cases, they are taken up using specific uptake mechanisms, but that again depends on the bacteria. But irrespective of that, they do come back into the cell. They bind to these specific transcriptional regulators shown here in green, and then they are able to affect transcriptional change. Now, to give you an example, now this is a very complicated system, and I've absolutely oversimplified it. But to give you an example of how the system would work, I go back to my favorite bacterium, Pseudomonas aeruginosa, which has not one but 3 different quorum sensing systems. 2 of them are shown here, and there is a hierarchy in this in how this quorum sensing system works. Pseudomonas. You have the LAS-I, which is the synthetase, which produces the quorumone. And like I said, in gram negatives, there are homoserine lactones. There are different kinds of homoserine lactones that they can produce. LAS-I produces 3-oxo-C12HSL. That binds to the transcriptional regulator in this case, which is LASR. So every quorum sensing system has a synthetase and a response regulator. In this case, the synthetase, which is the guy that produces the quorumone, is called LAS-I, which produces 3-oxo-HSL, which binds to the response regulator LASR. LASR then goes and does a bunch of things. There is a positive regulatory feedback. It enhances the expression of LAS-I, of LASR itself. A bunch of virulence factors. Most of these are secreted toxins and proteases. And we'll talk about secretion in a bit. But then it also goes on and turns on the expression of the second quorum sensing mechanism, which is the REL system for rhamnolipid production. And that, again, you have the synthetase here, the REL-I, which produces C4HSL, and the same process continues. And REL-R also turns on production of rhamnolipids, which are surfactants, very important for epithelial cell damage for the bacteria, a bunch of proteases, pyocyanin, which also cause epithelial cell damage, oxidative stress response mechanisms, including RPOS. Now, like I said, this also is an oversimplified process on how quorum sensing is regulated, because bacteria are, again, once they enter the body, they are under lots of stress to be able to colonize. And being a pathogen is not easy. The body has, over the millions of years, both the bacteria and the human body have evolved multiple defense mechanisms. So for a bacteria to colonize the infection, the human body, is not an easy process. So making your presence known by means of quorum sensing is a very important decision for the bacteria to be making, because what this does is it basically announces to the immune system that that there is an infection going on. So the bacteria do not take this decision lightly. What I show here is how complex the regulation of quorum sensing is in Pseudomonas aeruginosa, for example. Now, this is the LAS system, the REL system, and the third system, which is the PQS, which is a quinolone-based system. Now, there are multiple, all these blue boxes are transcriptional regulators, the green ones are sigma factors, and then you have small RNAs and RNA binding proteins. Now, all these come together and orchestrate the quorum sensing process, and the decision to so don't get put off by this figure. It's just to demonstrate how complex the regulation of quorum sensing is in bacteria. Even any virulence factor regulation is a very complex decision that the bacteria makes after considerable thought, if you can call it thought. Now, the other virulence mechanism that I want to touch upon is antibiotic resistance, which is a very pertinent process in bacteria. Now, there are multiple ways in which bacteria can resist antibiotics. The first one is not to let the antibiotic into the cell at all. And that it does by membrane impermeability. Again, going back to my favorite bacteria, Pseudomonas, it has one of the most impermeable outer membranes among bacteria. And that is because of the selectivity of the porins, which are present on the outer membrane, which is very selective and does not allow most molecules to enter into the cell. But even in those cases, some of the antibiotics can enter the cell, and the bacteria has other mechanisms to deal with the antibiotics that do enter the cytoplasm. One of the primary ones being target alteration. For example, DNA gyrase mutations are very common in response to DNA replication inhibitors, antibiotics, where the functionality of the protein is still maintained. For example, DNA gyrase can still go unwind and perform its helicase function, but it is now resistant to antibiotics due to several point mutations. You have efflux pumps, efflux systems, which are basically like pumps, pumping out the compound from the cytoplasm, reducing their cytoplasmic retention time. And these are typically tripartite pumps. The R&D efflux pumps are usually tripartite. What I mean by tripartite is that there are 3 components to it. You have an inner membrane component, which typically has ATPase functionality. You have a transmembrane component, which basically tends to hold them together. And then you have an outer membrane component. So what these together do is that in an energy-dependent process, they're able to selectively push drugs out from the cytoplasm, out into the external environment, that they're reducing the cytoplasmic retention time of the drug, and thus conferring resistance to the bacteria. Another type of virulence determinant that the bacteria have for antibiotic resistance are inactivating enzymes. Now, these are basically enzymes that are produced by the bacteria that specifically target antibiotic molecules and degrade them. An example being beta-lactamases, which act on beta-lactam antibiotics. Because the site of beta-lactam antibiotics are in the periplasm, because they specifically target the peptidoglycan biosynthetic enzymes, the penicillin binding proteins, for example. The inactivating enzymes, in this case, the beta-lactamases, are produced in the cytoplasm, and then they are secreted out into the periplasm, where they basically degrade the beta-lactam ring, rendering the antibiotic ineffective. Now, one of the ways, a very simple way that you can measure antibiotic sensitivity in the lab is by performing an MIC, where you have decreasing, so in this case, in an IDisc plate, you have decreasing concentrations of the antibiotics, the same number of cells which were input. You have the cell control here, and then you see you have 2 different strains, the wild type and a specific mutant. Right, so you'll see that the highest concentration of antibiotic, I'm sure you have read about all this, the highest concentration of antibiotic that kills the cell, that inhibits growth, not necessarily kill, is called the MIC. But then when you do this assay and you see something like this, you can clearly say that this cell is more resistant, this strain is more resistant to the antibiotic than this one, because this one requires more antibiotic to kill this particular strain compared to this one. Now, what you could do is that if you use specific subset antibiotics, like I've done here, so I've used specific MEX-CF OPRN efflux pump substrates, you'll be able to get very crucial genetic information using a very simple assay. So in this case, absence of this gene makes the cell more resistant to the antibiotic, which means that this protein, this particular transcriptional regulator AMPA, negatively regulates this particular efflux pump. Because as you can see, the MIC is drastically higher whenever you don't have the gene. So this again goes back into saying that with very simple experiments, you can get very meaningful information about the physiology of the bacterial cell. Now, I have spoken about secretion a couple of times when I say secreting this here and there. Now, that is mediated primarily by secretion systems. Now, bacteria, and they basically allow localization of the protein to wherever it needs to go, because due to the lack of compartmentalization, at least that we know of in bacteria, in terms of in the cytoplasmic content at least, all the protein synthesis happens in the cytoplasm, but then based on specific signals which are encoded within the polypeptide, bacteria can secrete. They are targeted to the secretion systems, and they reach their final location either in the periplasm or in the outside milieu. And that is mediated by secretion systems. There are 7 systems known so far, type 1 to type 7. Some of them are common, like type 1, type 2, type 3, type 5, which are specifically used to secrete toxins and most secreted factors, most virulence factors. Whereas type 3 and type 6 function as molecular syringes for direct toxin delivery into the recipient cell. Type 7 has been found only in mycobacteria till now and has not been found in other bacteria. And that has to do with the very unique outer membrane and the envelope system of mycobacteria with the mycolic acid and all that. Now, wait, why is this not switching? There you go. So this is a general architecture of the secretion systems, as you can see, which has a very similar concept to the R&D pumps, which is the efflux pumps that I was talking about. Basically, they form a conduit from the bacterial cytosol to the outside of the cell. So type 1, type 2, type 5 is not shown here, but both of these are involved in most of the secreted virulence factors to go from the inner membrane to outside the cell. Type 3, like I said, and type 6 too. We'll talk a little bit more about type 6 in a bit. But type 3, for example, is a directed molecular syringe-like mechanism which recognizes specific receptors on the host cell surface, and there is a toxin delivery, cytotoxins most likely, into the host cell surface. Type 4 is similar to the type 4 pili missionary, but we'll not talk about that. Now, type 6 is a very fascinating secretion system, which is developed exclusively for killing other cells. You have this harpoon-like structure here, and in response to certain external stimuli, the bacteria basically throw out this harpoon. It goes out, punctures the bacterial cell, and then you can have specific toxins which are secreted into the bacterial cell. Now, one important thing in all this, we can get carried away when we talk about bacterial virulence mechanisms, but then, and to realize how bad it is for human beings. But bacteria did not evolve these virulence mechanisms to target human beings, because they have been around for billions of years. Earth is what, 4.5 billion years old? And bacteria evolved, are thought to have evolved like a couple of billion years into the evolution of the Earth, into the formation of the Earth. So it's not like the bacteria got together like 2 billion years ago saying, you know what, guys, a couple of billion years down the line, these creatures called apes are going to develop into human beings. We better get things ready and start killing human beings right now. That did not happen. So bacteria evolved these virulence mechanisms in order to enhance their survival in natural habitats. All these secretion systems for targeted delivery, for example, were developed to overcome other bacteria, to avoid amoebal predation, because those are hostile environments like soil and natural habitats of the bacteria. Everything out there is out to kill them. So bacteria have evolved survival strategies in order to survive. And humans just happen to be chance hosts. Granted, there are some virulence factors which seem to be specifically targeted to human beings, and that's a result of niche evolution. When the bacteria adapt to a particular niche, then again, like it exchanges genetic information with local microbial communities, and it adapts itself. Now, a case in point I'm going to talk about is the type 6 secretion system. Now, John McLanus's group at Harvard showed in a very elegant experiment. I'll show you a little bit of a small video clip in a bit, but a little bit of background before that. What you will see in the following clip are two different bacteria. The Vibrio cholerae bacteria are tagged red with the RFP, and the Pseudomonas aeruginosa cells are tagged green with the GFP. Now, what you will see in the video is that the Vibrio cells, now the type 6 secretion system is also tagged red. So when you see a bright red spot, it means that the Vibrio cells are expressing type 6 secretion systems. And then when you see a bright green spot, it means that the Pseudomonas cells are going to produce a type 6 secretion system. So what I want you to watch for is that the initial attack of Vibrio cells to Pseudomonas has immunity proteins to the type 6 toxins, so it will not get killed. But then in a retaliation attack, Pseudomonas cells are going to start forming type 6 secretion systems. And as soon as that happens, as soon as you see a green spot, you'll see that the red cells are going to round up and die. I want to play this quick video clip. Again, you see the red cells and the green cells. Whenever you see a bright green spot or a bright red spot, it means that the type 6 secretion machinery is being fired. Watch closely to the red cell as soon as there is a green cell firing. You will see that it starts to round up and die. There again. All over. So this is a very fascinating finding, and what this reiterates is that, and all this happens in a time span of 2-3 minutes. But what this reiterates is that these virulence mechanisms were to enhance bacterial survival, like in the environment, where killing other bacteria allows this bacteria to have better access to resources and everything. I'll not go into antibiotic resistance as a suffice to say that the same concept applies there too. But what allows these bacteria to be such successful pathogens is that they have a very plastic genome. And what that means is that the bacterial genomes have great flexibility and adaptability in terms of being able to take in new genes from the local environment. And so what that means is that there are two basic sets of genomes. You have the core genome, which is the ancestral genome present in all strains in the species. And then you have acquired genomes, which are acquired genes, which are which the bacteria take up by horizontal gene transfer or from transposons. And this allows adaptation of the bacteria to the specific niche that they are there. These can be virulence genes, and in many cases, they can take up large chunks of genes, a few kb, few hundred kb even, and they are localized in pathogenicity islands. But what all these come together to do is to regulate virulence factor expression, which is a very complex process, like I was telling you, which involves transcriptional regulators, sigma factors, and regulatory RNAs. And the expression, again, is timed in response to external cues, the signals that the bacteria receives. Now, we don't know a lot about a lot of these signals that the bacteria receives, and people are trying to find out what they are. But some of the mechanisms by which, now, some of the mechanisms by which the bacteria recognize these signals are by using two component systems, which form the eyes and ears of the bacteria. So they basically recognize and bind external signals. And there are two components to this, as the name suggests. There is a sensor histidine kinase, which has an effector binding domain. And then you have a phosphorylation domain here. And then the second component are the response regulator, which are the transcriptional factors affecting the transcriptional change. In response to a signal binding, there is a phosphorylation event that then, because it's a kinase, it can cross-phosphorylate. These are basically dimers. But then the phosphate group is transferred to the response regulator, and then the response regulator can go in and affect change. So what this means is that in response to a particular signal, bacteria can and do change their transcriptional response as an adaptive mechanism. There are many variations to this, but we don't need to get into those variations. Basically, they form the eyes and ears of the system. And in addition to transcriptional regulators, you also have non-coding RNAs or small RNAs, which mediate post-transcriptional regulation of not only virulence genes, but they affect bacterial transcription in general. For the most part, these are antisense RNAs. You also have cis RNAs. Antisense RNAs are mostly transacting, and they can either repress activation of the mRNA by binding it, causing double-stranded structures, which are basically cleaved out, or they can activate gene expression by exposing ribosome binding sites by binding to them, activating gene transcription. Another common theme that you see in many different bacteria to regulate virulence, small RNA mediated, is these small RNA binding proteins. They're known by different names. For example, this one is called CSRA in E. coli, Vibrio, and other Enterobacteriae. In Pseudomonas, it's called RSMA. But the concept of how it works is the same. These small RNA binding proteins bind to mRNA, affecting their stability. Translation is affected. But small RNAs, such as RSMY and RSMZ, or CSRA, CSRD, they can bind, sequester the small RNA binding protein so that the mRNA is free to express. Now, like I've been mentioning all along, these are very complex processes of small RNA gene regulation. The bacteria does not take it lightly. So this is an example of how virulence genes are regulated in Pseudomonas aeruginosa, where you have numerous transcriptional regulators. So what you see in this figure are the orange boxes. I don't want you to be overwhelmed, just to drive a point that the process is very complex. There is multiple redundancies present. There is all these orange boxes are transcriptional regulators. You have sigma factors like ALGTU, RPOS, all those here, for example. You have small RNAs, like you have RSMY, RSMZ. These are the small RNA binding proteins I was talking about. You have more small RNAs here. All these come together to coordinate expression of virulence factors, which are shown here in blue boxes. There are, like I said, Pseudomonas aeruginosa is a multifactorial virulence factors. So there are multiple virulence factors which are spatiotemporally expressed, which together contributes to the virulence of the bacterium. And again, this is a very exquisite method of regulating expression. There are multiple layers of redundancies because the bacteria does not want to regulate or express virulence factors lightly. So there are multiple levels of redundancies, and what this also allows is for the bacteria to very finely tune expression in response to various signals. Now, how do you go about to understand virulence once you're starting? Now, you could take the basic forward genetics approach, like what Mendel did. He did this green and yellow round and wrinkled piece. So he saw a phenotype, and then he went on to understand the genetic basis of it, right? Or at least he tried to. So you could do the same approach here. Once you have a virulence phenotype and you want to study it in the lab, what do you do, right? You can also do a reverse genetic approach, but again, this is a general outline of the techniques that are involved in the process. It's not necessary that you have to do it in this same order or even use all the techniques. All this has to be tailored to fit your approach. But let us say, given those caveats and those, let's start with this. So I'm going to quickly finish off in about 5 minutes or so we can have some time for questions. So let us say you have a virulence phenotype, right? So, and you want to identify the genetic basis of it. What are the other genes involved in the process? How does it affect the physiology of the bacterium? You can start off with the TNC. Now, I'm going to quickly go over the protocol for TNC and how it is done. So what you would do is basically a transposon library generation. So the concept here is that transposon insertion into a gene inactivates the function of the gene, inactivates the gene is no longer able to function. So what you would do is that you would make a library of strains. You would take your collection of cells, and then you will treat them with the transposon, typically expressed on a plasmid, or you could even transform it directly. What that's going to do is that the transposon is going to go randomly insert across the genome. And there are ways of saturating the genome in such a way that there is a single transposon insertion in every single gene of the bacterial cell. At least that's the ideal situation. Once you have that, you can do a screening process to, for example, if the phenotype you're interested is in biofilm formation and you want to identify all the genes that are involved in biofilm formation, you take a transposon insertion, a random insertion library, and do the biofilm formation assay. So there are two scenarios here. One, you'll be able to identify activators of biofilm formation in the sense that when there is an activated disruption, the cell will no longer be able to form a biofilm, or you can have repressors of biofilm formation where if there is an insertion in the repressor, the cell is going to start more biofilm. So when you take the library and you do the biofilm assay, you'll then look at variations across the spectrum. Is this whether the cells are going to form low biofilms or higher biofilms? And once you have identified those guys, those specific clones, you'll be able to do a next-gen library preparation, sequence it, and then you can identify where that specific gene has been inserted. And once you do that, you can then, once you identify the genes in the process, you can do a bunch of things. You can do a large number of in silico analysis, including BLAST, homologs, ortholog searches, locus analysis, which is a convenient thing that you can do in bacteria, because in many cases, not necessarily most, in many cases, bacteria tend to cluster genes involved in a specific function of the same locus. You can also do protein motif analysis and a bunch of other in silico analysis. But once you have identified the genes, you can also do isogenic mutants to try and understand how that process works. So what you would typically do in an isogenic mutant is, let us say you want to delete just gene of interest. You would amplify the upstream and the downstream region. This is not the only way to do it, but this is a widely used method. You would amplify the upstream and downstream regions of the gene, leaving out the majority of the gene, and then you'll splice them together by PCR or by cloning, and put that onto a suicide vector. And then you can use this suicide vector to do allelic replacement by recombination, in the sense that you replace the wild type copy of the gene with a mutant copy of the gene. So when you say a deletion of a gene, this is what you mean. So your wild type copy is replaced with a deletion copy. And once you do that, you can, after confirming it, you can do a bunch of different assays. You can do RNA-seq to look at the transcriptome, how deleting that one gene has affected the transcriptome of the cell, how deleting the gene has affected gene expression across the whole thing. You can look at phenotypic microarrays, which is basically hundreds of thousands of phenotypes that you can do in 96 well plates. You can look at how it affects the proteome, the metabolome, or if it's a transcriptional regulator or anything that binds DNA like HNS, you can do something called a ChIP-seq. And I think it's about ChIP-seq quickly. We'll come back to ChIP-seq in a bit. And then you can also do phenotypic assays depending on what genes are being transcribed or what you find in your in silico analysis. You'll get a general idea of what that gene is involved in, what virulence factor the gene might be involved in. You can do in vitro assays based on that. You can do animal studies based on that to look at various stages of colonization, whether it's early or late, or whether it's toxin production. And then finally, you can do network analysis. You can either do in silico analysis like a string analysis, or you could have a comprehensive network of transcriptional factors and virulence factors and see where your gene of interest fits into that process. So this is a typical data set that you get from RNA-seq, where you basically, what an RNA-seq does is that it looks at the transcription across the entire genome, where each of these lines represents the expression pattern of a specific gene. So what this allows you to do is that you can see which genes are overexpressed and which genes are repressed under whatever condition you want to do. ChIP-seq is again an elegant technique where if you want to look at a protein binding to the DNA, you can do a tag, an epitope tag to it. You can cross-link the interaction with formaldehyde. You lysin sonicate. So the basic idea is that once you do this, you can do an immunoprecipitation, pull down the specific transcription factor that you're that you're interested in, and then you can get the DNA out that was protected by it and do a next-gen sequencing. So what all this will do is to help identify the genetic network, to try and get a better understanding of, we don't need this. This is an example of a string. So this basically helps in identifying what the network it is involved in and what are the various virulence factors. And all this will come together in trying to design intervention strategies for to deal with antibiotic-resistant bacteria. So in conclusion, I want to emphasize that virulence factors, it's not a host-centered concept. It is a very complex and dynamic phenomena involving both the microbe and the host. And studying this interplay will help elucidate underlying mechanisms that are involved in the process and help in designing effective treatment strategies. I want to thank you all for listening. Thank you for inviting me. And I'm open to questions if you have any. Thank you.
Dear participants, you can post your questions. You can post your questions. You can post your questions in the chat box. So Dr. Deepak would be happy to answer them.
So Professor Rajarajan, how's this going to work? Are you going to ask me the questions or?
Yes,
Yeah?
it
Okay,
is.
okay, great. Because I'm not sure how, oh, there you go. Perfect.
There is a question from Dr. Anant Krishna.
Uh-huh.
He wants to ask you, are the pheromones secreted through the secreted pathways 1 to 6, similar to the killing mechanisms?
Yes, they are. So not necessarily the secretion pathways. They are also secreted through the type 2. But there are also efflux pumps, like the MEX-CFOPRN I was talking about, the MEX-AB pump. So there are multiple different pumps which secrete the pheromones, but also secretory pathways. But yes, the short answer is yes.
There is one more question. What is the overexpression after isogenic mutation?
So it's not necessarily both. So in some cases, you cannot delete a gene because the gene is essential for function of the bacteria. So you delete the gene, the cell is going to die because there's no redundancy there. So in those cases, you cannot make isogenic mutants. You have to make an overexpression constant or a conditional mutant. So what I mean by that is that you'll clone the gene on the plasmid. So it's the inverse phenotype. What you're doing when you're making a deletion construct is that you're going to delete a gene and look for absence of function. But when you make an overexpression construct, you're going to look for gain of function because a cell that is going to form a gene that is responsible, an activator of a biofilm formation, for example, is going to hyperproduce biofilm when you overexpress the construct. So you can do both if you want to, but typically people do one or the other, either a loss of function or a gain of function.
Okay. There is another question, another participant asking a question like, what should be the concentration of sugar during culturing of biofilms?
It depends. If you're using a rich media like LB or tea broth, for example, you don't need to bother about that. Your concentration of sugar only comes into question when you're using minimal media, a chemically defined media. And there is no straight answer to it, but generally about 0.5 to 1% carbon source typically works. But again, it depends on the bacteria, depends on the culture conditions. If you want to look at the rich media, you don't really need to add more sugar, like I said, because there's enough carbon source in the ingredients that you add, because they're typically complex, like peptone or tryptone or whatnot. So your sugar concentration comes into play only when you use chemically defined media, and it's typically about 0.5 to 1%.
Dear participants, we invite a few more questions. There is another question coming up stating, what is the recalcitrant, what is the recalcitrant infection mean?
Okay. So recalcitrant means that it's just very resistant to antimicrobial agents. And it's very difficult to get rid of recalcitrant infections because of the properties, the physiological properties of the bacteria. For example, cystic fibrosis. In a cystic fibrosis lung, Pseudomonas, Burkholderia, staph, all these can infect your CF lungs. And you're infected for life. Once you get infected, that's because of the conditions that are present in the CF lung. There's enhanced mucus. There is some amount of conditions that favor bacterial colonization. And those conditions are recalcitrant in the sense that you can never get rid of the infection. So the patient remains infected for life. So throughout their life, they have the bacteria in their lungs. And that's an example of a recalcitrant infection. Other examples, for example, would be deep wound infections like Clostridia, which are also recalcitrant. And the property of recalcitrance comes from resistance mechanisms, antibiotic and antimicrobial drug resistance, which makes them difficult to eradicate.
All right. The next question is, what about the virulence of saprophytic bacteria? Do they emulate the similar mechanisms and conditions?
Yes, they do, and they do not. As in, saprophytes are primarily opportunistic pathogens. So in the sense that they don't have a single virulence factor contributing to it, and there's a lot of underlying factors that need to happen, that need to be present for the saprophytic bacteria to cause a disease. So again, they have to be immunocompromised, or the specific virulence factors that they possess, which will not work in a healthy host, will then be able to work in an immunocompromised host, or under underlying conditions which will enhance infection process.
Yeah. The next question which came up is the TC sequences possible for all bacteria?
I'm sorry, what's it again? What sequences?
TC sequences.
DC?
Sequences are possible for all bacteria to be done.
I'm not sure what DC sequences, what they're referring to.
T for 10.
Okay.
TC sequences.
TC sequences. I'm not sure what they mean. Maybe they can elaborate what TC sequences mean.
Sure, I'll come back to the question.
Yeah, so whoever asked the question, if you clarify what TC sequences mean,
I'll come back to it again.
Yeah, of course.
In the meantime, the next question is, can bacteriophage mechanism be used for COVID drug discovery?
COVID drug discovery and bacteriophages. I'll have to say no, because bacteriophages are specific to bacteria. COVID-19 is not. So using bacteriophages for drug discovery is going to be unlikely. I'm not sure if they're referring to CRISPR-based mechanisms, but because CRISPRs are derived from bacteria and bacteriophages, but at the outset, I'll have to say no. I don't think so.
Yeah.
Maybe there's some out there that can come up with a way to use phages for COVID drug discovery, but I'd have to say no.
The next question is how to identify a functional gene functional gene of these sort, like those functional genes which contribute to quorum sensing and which do contribute to virulence factors and so on in a bacteria. How to identify them?
So identifying genes responsible for virulence factors is the question, right? So like I said, the TNC could help because if you do a random transposon mutagenesis, and you have so the success of TNC, which is the primary way to identify, so if you're going from a phenotype down to the genotype, like the forward genetics approach, a TNC is the best way to do it. That would be the most straightforward way to identify on a whole genome scale what are the different genes that are involved in the process. Otherwise, it gets more labor intensive to do this process. But if you're doing a forward genetics approach, in the sense that there is a gene that you're interested in and you want to identify what role it plays, then you can do go straight for a deletion or an overexpression construct and do a bunch of different phenotypes that you're interested in. Hopefully one of them would click, and then you can go do further assays from there. But if you're going from a phenotype and you want to identify what are the genes involved, the TNC is the most direct approach that you can perform on a whole genome scale. Yes, so.
Yeah. According to you, which is the most effective regulatory method in the bacteria you find?
Most effective regulatory method? Well, I'm a bit biased towards because I think that's a very way of expression. And why regulatory RNAs is because, see, bacterial mRNAs have a very short half-life, right? Because they don't have the poly A tail, which helps stabilize them like eukaryotic RNAs. So what small RNAs do is that they allow for very rapid changes. Like in a few seconds, you can have expression of a particular gene. And that is brought about by regulatory RNAs, because what they do is they can do a gene silencing, keep the gene, so the mRNA is transcribed, and then the small RNA will go and silence the gene. And in that sense, as soon as the cell receives a signal, it can start expressing right away, which allows for much more, a much more rapid because they have so much response of
the next question by another participant
yes, that's
is,
a very,
can we use molecular
interesting
syringe
question,
mechanism as a
very good
potent drug
question.
delivery system?
Yes, the short answer is yes. People have looked into it. In one of the neighboring labs where I was working, they actually used this, the type 6 system from Pseudomonas. So there was this person from Novartis who was working there, and she developed the system to use isolated type 6 secretion machinery to kill other bacteria. A very fascinating story. It came out in PNAs, I think, in 2015 or 2016. You should go look it up. The name is Elena. But yes, anyway. But yes, the short answer is yes. A very good question, yes, but you can do it.
Going back to an earlier question, I guess it is not TC, it is TN sequence. Yeah. So TN sequence, is it possible for all the bacteria of this sort?
Yes, you can. Yes, of course, yes. Because it's genome independent, and it's platform. The sequencing is platform independent. Initially, you used to be able to require genetic information of the bacteria to be able to do a TN-seq, but now you don't have to, so long as you know the general category of bacteria, and that is just to know what transposon to use. But generally, most of these tools have been very well developed. So you can have a TN-seq for where you don't have any genome information. You can still be able to do it. So TN-seq, RNA-seq, ChIP-seq, any of those, all of these are, you don't need to have any sequence information of the bacteria. And the sequencing process is also sequence-free. It's platform independent. So short answer, yes, you can.
Coming up next is a question whether the functional active gene differ from different strains of same species.
Functionally active gene is a different.
Functional active gene, do they differ from different strains of the same species? Probably
Yes,
he or she is referring to a serovar.
Yes, yeah. I mean, there is, for the most part, if it's a critical regulator or a gene involved in a critical function, it is conserved throughout. And that is one of the things that we look for when we do, when we identify a gene of interest. That's one of the first things we do. We do a homology search to see how conserved the gene is across the different bacterial species. So there are different degrees of conservation. You'll have genes that are conserved across different bacteria, across different bacterial genera, and those are ones that are involved in more fundamental processes of the bacteria. Whereas when you have genes that are more specific to that particular strain or to that particular species, then there is further less conserved nature of those. But in many cases, if it's a critical regulatory system, then it's conserved across many different bacterial species. But you do have minor SNPs, single nucleotide polymorphisms, which allow for fine-tuning of the expression process or fine-tuning of the function. So, yeah.
Yeah. The next question is it possible to use antisense RNA technology to develop a vaccine against COVID-19?
Yes, it's possible if you can find a way to deliver antisense into the COVID-19 virus, because the technology exists to. I mean, getting an antisense sequence is not a big deal. You know, you can, once you have the genome information, you just synthesize. It will take you a couple of days to get the sequence.
Yes.
But the problem is with the delivery. How are we going to deliver the antisense fragment into the cell? Now, you could have a transgenic cell. You could package it into a retrovirus and have it infect the cell so that when it infects the human cell, the replication is stopped. There are ways of doing this, but then understand that these are mechanisms that require a lot of research. This is not something that can be done overnight or even in a few months. These are things that require a lot of standardization, a lot of work needs to go in before you can even have the technology ready for specific pathogens. And then furthermore, to develop that into a vaccine, to determine the efficacy. So in the long run, yes, it is possible to do it. Antisense works great in many different diseases, but the time frame to get there is going to be, it's not going to be short.
Are there chaperones present in all these types, and how critical are they?
I'm sorry, are there chaperones in what?
Molecular chaperones. Are there chaperones present in all these types of strains, and how critical are they
Yes.
in contributing certainly to the virulence?
Virulence, yes. So good question. Chaperones are fundamental to bacteria, because any sort of stress that they receive, chaperones are the ones that deal with misfolded proteins, for example. Like I said, the host is a human host, or any host for that matter, is a very stressful condition for the bacteria. So there's going to be a lot of stress involved, RPOS upregulation, and there's a lot of misfolded proteins. So chaperones are an integral part of the of the adaptive process of the bacteria. So they're not involved in, they're not specific to any one virulence pathway, but they are critical for the general survival of the bacteria under stressful conditions, to deal with misfolded proteins, to deal with various other stress situations. So yes, they are integral to survival as a whole, and they have a contributing effect, but they're not generally considered a virulence factor for that. Because they help in the adaptive process, but they are not by themselves a virulence mechanism.
The next question is, what is the latest virulence factor identified in Pseudomonas?
Latest virulence factor, well,
Your favorite bacteria.
my favorite bacteria, yes. So a very fascinating story. It's not a virulence factor by themselves, but so this is a marriage of two different of my favorite bacteria, phages and bacteriophages and Pseudomonas. So Pseudomonas has a lot of prophages. Prophages are silent phages that are part of the bacteria, of the bacterial genome. And the case in point are PF1, they are filamentous phages. Now, what they do, and people did not know what those phages were doing there in the bacterial cell, but a paper that came out last month, if I'm not wrong, has shown that under certain stressful conditions, I mean, we knew that under stressful conditions, bacteriophages, prophages are induced. But what these people showed is that these prophages, after they're induced, they are filamentous phages. So they basically go cover the cell, they form like an armor around the cell and do not allow antibiotics to get in. I found that mind-blowing. That I mean, granted, bacteria had millions of years to evolve these systems, but then the whole concept that bacteria carry these phages as an armor, they secrete under certain conditions, that goes and covers the bacterial cell in addition to all the other mechanisms, and that protects the bacterial cell against antibiotics was amazing. And I'm sure that also helps in immune evasion, but they've not studied that yet. But yes, so
Yes,
that's
yes. Yes, it is. Yes, it is. One last question, Dr. Deepak. Can any other gene be used for silencing gene instead of a transposon? Will it not flip again back because it's basically a jumping gene?
Yes. So the workaround there is that you do not give it a transposase. You have only the integrase, but you do not have the excisionase in the system. So yes, it's a very valid point that they're jumping genes, they're going to jump around. But when you introduce the transposon on a plasmid, you're going to have only the integrase. As you know, the excisionase is the one that's required for taking it out. The integrase is the one that's responsible for integrating it into the system. So you will not have the excisionase on the plasmid. So it's going to, once it goes in there, it's going to remain there. And that's the workaround for that.
Yeah, I guess we are done with the Q&A session, Dr. Deepak. I guess so.
I just
There are
want
no more questions.
I hope that this was helpful. If you walk away learning at least one fact about bacterial virulence mechanisms, I'd be happy about it. And good luck to you all, to all the students.
Certainly. In fact, there are quite a lot of, quite a lot of participants from the teaching fraternity as well.
Oh, okay.
Yeah. So okay. This whole entire academic participants do not include only the students, but I mean, you have quite a lot of researchers, quite a lot of people from multidisciplinary field who are some way or other directly or indirectly related to the bacterial virulence mechanisms. A lot of teaching, lot of people from the teaching fraternity.
Okay.
They have really interested a lot of weight on the webinar.
Oh, that's good to know. I wasn't aware. That's good to know.
Because a sizable number were teachers.
Okay.
Yeah.
Okay.
Anyways, coming to the concluding remarks, My heartfelt thanks to our chairman, a benevolent leader, Dr. Somshekhar, and Madam Secretary, Mrs. T. Bharti, who are always a driving force behind all our academic endeavors. I'm extremely thankful to our COO, Chief Operating Officer, for his constant support ever. My sincere gratitude goes to our beloved principal, Dr. E. Jerome Xavier, who is always a pillar of support in any academic endeavors we venture into. My deep sense of appreciation to you, Dr. Deepak, for your spectacular, highly informative, and enlightening presentation. I'm immensely impressed with your talk, wherein a participant from a non-microbiological background could even understand the concept. Thank you, Dr. Deepak, for that. I'm one for who always believes in teamwork. Today, you all have witnessed how teamwork can be highly successful. Kudos to my dedicated faculty team, without whom today's webinar was not a possibility. As well, my sincere thanks to Mr. Chandan, our IT manager, who was always there with us when we needed him the most. And finally, I would like to thank all the participants for the smooth conduct of the webinar and wish you all a very best in all your future endeavors.
Thank you, sir.
Dr. Deepak, thank you very much. It was fantastic presentation. Yes, I know the students will enjoy, the teachers as well. It was very good.
Thank you. Thanks for that. And thanks for the opportunity to share my ideas with you guys. It was good. These sort of outreach programs are very important, and it's a great initiative that you guys are doing in having the sort of guest as a way of different perspectives on age-old topics. So it's very nice that you guys are doing this, and I appreciate you for that.
Good.
The pleasure is ours.
Thank you, sir.